Ladies and gentlemen, good morning and afternoon. Today is the fourth education webinar in this series by which we are providing customers idea how to transition their research to a GMP production setup. I am the host of the event today, Jeffrey Poon. I am the commercial manager of Biosafety Carbonate, CO2 incubator and biological shakers in APAC. I am based in Hong Kong. Today, the title of the talk is GMP Requirements for Cell and Gene Therapy Institutions. The speaker will be Chris Wronski, our cell culture application scientist for EMEA. He's going to give us a talk from UK, which is 8 hours behind us. He will be giving us a mental process, what we need to prepare to set up a GMP facility, using an example where CO2 incubator will be used for gene and cell therapy. Chris is a cell culture application specialist with Thermo Fisher Scientific, where he provides expertise on cell culturing applications for both internal colleagues as well as scientists around the world. His knowledge is based on his combined 14 years of experience in academic research as well as pharmaceutical research. So he worked for Covance before, so where he has got a strong knowledge of about how to work with a GMP setup. His scientific background comprises molecular medicine, microbiology, immunology, epigenetics, development of cell therapy for brain metastasis, as well as developing a wide spectrum of characterization methods for varied therapeutic antibodies. Chris holds a master's degree in biotechnology from the West Pomeranian University of Technology in Poland. Before the start of the talk, please allow me to give you some suggestion. So there is a button on the bottom right corner where you could post your questions. I suggest you to send your questions to panelists if you prefer not to disclose your identity. Now, during the talk, you will be muted to ensure the smooth proceeding of the talk. The talk will last for about 45 minutes, after which we will have the Q&A sections. Now, without further ado, please welcome Chris. Chris, the stage is yours.
Thank you so much, Jeffrey, for introducing me. Can you hear me well?
Yes.
Fantastic. Okay, so let's start my presentation. Hi, everybody. Thank you so much for attending my presentation. Dedicated to GMP requirements for cell and gene therapy institutions. And I'm not going to introduce myself again, as Jeffrey did an absolutely great job. Thank you so much, Jeffrey, for that. So let's go to first slide. Okay, so I'm going to stop the video now so you can guys concentrate on slides and on my voice. Okay, so as Jeffrey indicated, firstly, I would like to briefly explain why I was appointed to give you actually this webinar. Before I have joined Thermo Fisher Scientific, as Jeffrey said, I worked approximately about 14 years as a scientist based in a labs in UK. I spent about 6 years working in academic research. I was involved in multiple cancer research projects, with the last one actually dedicated to developing new cell therapy for the brain metastasis. And I also spent a few good years working as a method development scientist for a global pharmaceutical company, Covance. And I was based in the team responsible for developing a range of characterization methods for varied large therapeutic proteins like monoclonal antibodies. And the methods had to be routinely run in a GMP labs by the analyst. And I must admit to you guys that that transition from academic lab into GMP lab, it was very, very challenging for me. And I am here really to share with you my experience. So let's look at the agenda for this presentation. So firstly, I will try my best to explain what good practices are. Terms like GMP, GLP, and GCP, what they mean. Then I will talk about different key elements important for the lab to function and how they differ between academic labs and GMP labs. We will also explore on the cell and gene therapy institution's needs and talk about the biggest challenges. Then we will also analyze step-by-step equipment acquisition. And finally, I will also talk about how to choose the equipment accordingly with different GMP principles. So I think the best method to explain and visualize the good practices is to show it along with drug development process, as different types of good practices must be implemented and followed in different stages of the drug life cycle. So starting from the top left of this slide, as you see, basic research and drug discovery are not controlled by regulation at this early stage. Simply because in this stage we don't really know yet if particular molecule or therapy will enter further stages. And also in these early stages, testing safety of the drug on a living organism is not required yet. But when the decision is made that particular therapy or drug could be utilized, beneficial for the human use, then we must to progress to that testing stage. And in the first stage of preclinical testing, or called preclinical development, or sometimes called early development stage, in most of the cases, live animal organisms are involved, and the safety of the drug is evaluated. This stage is controlled by GLP, or good laboratory practices. And the main goal of these good practices is to protect scientific data integrity. In the second stage of testing, clinical trials, can be a number of those trials, the human subjects are involved, and therefore this stage must be also controlled by different ethical standards in form of GCP, good clinical practices. In this stage, safety and efficacy of the drug is assessed. Successful completion of the testing can then result in regulatory approval, and company can start producing, manufacturing on bigger scale. That production of the therapy of drug is controlled then by GMP, good manufacturing practices, to ensure consistent quality of the product. The common term used is also CGMP, or current GMP, which means complying with the latest processes, up-to-date technologies and systems. If you notice as well on this slide, for all the good practices to be super effective, they must be accompanied by good documentation practices, GDP. As without effective, those extensive documentation, this whole drug life process could not be accurately controlled. So therefore, as we see on this illustration, on this slide, GMP are all about producing safe and pure and high quality product, and this can be successful through effective implementation of all GMP elements, principles. GMP are also minimum requirements that manufacturers must meet, and what we also really need to remember, and it is actually very important, that GMP are only a guidance, not rules. Therefore, we have multiple ways to comply. This can be very different for different manufacturers. As we already mentioned, good documentation practices are extremely important, and second crucial element to ensure that successful implementation of GMP is also quality management system. We will explore on that later in this presentation. So let's talk about the differences between academic labs and GMP labs. This hopefully will help you to understand GMP guidance better. So as you see on this slide, the main determinant for all the differences between academia and GMP lab is the final product or the final goals. In the academic research, as you see, it's intellectual property or expanding our knowledge And in GMP lab is production of drug therapy. The product in GMP is directly connected with patient safety and patient life, really. So as you see, there is a huge responsibility and a lot of risk if the manufacturer produces low quality therapy and consequently can risk patient safety. So let's discuss the key GMP critical elements like documentation, quality management, and equipment first. So the main reason for documentation to exist in GMP is to ensure that traceability. It is important to be able to identify the reason for the decreasing in quality, for example, and be able to respond as fast as possible to fix the problem and prevent it of occurring in the future. And extensive control documentation allows to identify any of those potential problems. In academia, control documentation is not really required as no patient safety is at risk. For the same reason, quality management is also not required in academia. But in GMP, that quality management is again extremely crucial as to ensure the product is fit for intended use and again, safe for patient. The equipment plays enormous part in this equation. As you know, almost every step of the process involves various equipment. Therefore, equipment in GMP must to support GMP compliance. So again, the product data on manufactured product, produced data, manufactured product is always consistent and highest quality possible. Other key elements I would like to discuss briefly are design of the lab facilities, training, and sample management. The design of GMP lab facilities need to allow all the tasks flow efficiently, and what is super important, the design must to assure the lowest or minimal contamination risk to the product, and this is a priority. In the case of biologics, cell and gene therapy, the production must to take place in a clean rooms where the particles in the air are controlled and filtered out in that aim to reduce that contamination risk. And the equipment has to be also compatible with the clean room, which means it have to be designed in a way so it doesn't generate too many particles during the normal operation. In academic lab, student safety takes the priority. Next key element, training, also plays crucial part in GMP. The training, especially for new employees in the lab, is usually very structured. It can take easily up to 6 months. Huge part in this training plays documentation and quality control again. I must to say, I never forget the pipetting training I had to attend also when I joined GMP Lab. It was 1 week long and I had to pass that training. And you can imagine, despite having years of pipetting experience in academic labs. So the equipment plays huge role in the training as well. As you can imagine, some equipment can be designed in a way so it's easier to train on. And the main goal of that structured training in the GMP is so everybody work is super consistent and uniform. Nobody's really allowed to develop personal habits in GMP labs, as opposite to academic lab where, as I remember, everybody had its own habits. The third key element and last one I want to mention is sample management. The global pharmaceutical company Covance I used to work for invested a lot of effort into control of the samples. And I'm really not surprised. We had a sample management system where all activities on the samples had to be recorded at the actual time of this activities taking place. Plus, we also had a sample management team, full team designated to sample processing. All that as samples in GMP, as most of you know, most of the time have huge monetary value and additionally reputational value as they come from the clients or patients. And in academia, samples are also important, but for different reasons, as they may, for example, high research value. Okay, guys, I have 3 questions in this presentation for you, and I believe you can participate and answer the questions in a chat box. I want to test in this presentation how much you learned, and just to engage you and just break the presentation to make it more interesting. So the first question, guys, is a very tricky question, as all the answers firstly may be for you pretty much correct, but I want you to choose one most relevant reason for GMP to exist. What is that biggest priority in GMP? And the answers are A, documentation and safety, B, quality control and high quality product, C, to produce high quality, pure and safe product, and D, regulatory approval and safety. Okay, I can see the answers coming already, and I can see the correct answer. Yes, you're absolutely right. The first priority for GMP guidance to exist is to always produce the high quality, pure and safe product. This is a #1 priority. Thank you guys for participating. Okay, let's talk now about cell and gene therapy institutions' needs and the biggest challenges. So cell and gene therapy needs are quite specific and very much connected with different phases or stages of therapy development. In the research discovery stage, usually wider performance range and highest number of innovative features are usually preferred. And also, as you probably know, this is also connected with the complexity of the applications you run. Then when we enter that stage of preclinical, clinical testing and production, basically that region, we talked before of safety testing and manufacturing controlled by those good practices, these needs are changing really dramatically. The needs are dictated by regulatory requirements, documentation, certificates play crucial role. The priority is monitoring, as I circled here in the slide, traceability, the ability to document and quality check every single step of the processes. Therefore, durable, well-built, easy to clean, easy to train on, easy to operate equipment is more desirable in GLP, GCP, and GMP environment. Okay, so the biggest challenge is overwhelming number of required documentations. And this always gonna stay with me as documentation was really huge part of my job when I used to work in Covans. The ones needed to be supplied with equipment by equipment manufacturer, and all the critical standard operating procedures to be created and implemented by cell and gene therapy institutions. And don't forget about updating and maintaining all those SOPs on regular basis. Due to variety and high number of documentation needed, many institutions, especially these small startup companies, are confused what is needed and what are actually your responsibility in terms of documents. Second challenge is quality management. Correct implementation and control of all quality system elements is also super challenging. Quality control, detecting, preventing quality issues should be everybody's responsibility, really, in GMP. And evaluating the risk to quality should become also every worker's second nature in GMP. And of course, let's not forget the challenges with acquiring the right equipment, right? The confusion can be enormous in terms of what is required. So let's discuss these 3 challenges in a bit of more details. Okay, so we need to understand first the types of documents. And the type of documents and how they really connect with the equipment. So I try to divide them into 3 categories. First, the documentation provided by supplier, like Thermo Fisher Scientific and what we as a company should provide to you guys, so you can comply and pass the regulatory audits. Generally talking, these documents must to serve as a proof that equipment or individual parts are tested for intended use. The types of documents can be rather specific for different type of equipment and can be specific as well depending on your validated processes. Therefore, Thermo Fisher Scientific have to be flexible in these terms. I will give you examples in the next slide. So the second category of documents are your responsibility really to produce, maintain and update, and are associated with your unique processes. They are used to give precise instruction to your analyst in your GMP lab. They are used to document every step in your operation. Most often they are closely linked together. Examples are, for example, written procedures, working documents, reagent preparation sheets. And the third category of documents are also your responsibility to generate and maintain, and they are directly linked to equipment usage, cleaning, and maintenance. Examples can be usage and calibration logs, day-to-day usage instructions, general and specific cleaning, and even list of cleaning agents. They are produced by you, but Thermo Fisher Scientific supplier can make that job easier by providing you with comprehensive user manual, recommended cleaning procedures, or compatibility certificates with different cleaning agents and procedures. So here on this slide, I wanted to show you examples of GMP compliance documentation provided by Thermo Fisher Scientific with our CTS cell therapy systems equipment. As I mentioned before, the content can vary depending upon the equipment purchased and your requirements. Examples are replacement parts list, cleaning procedures, different really types of certificates, factory acceptance test, report known as factory acceptance test report, design qualification documents like technical data sheets and drawings. If the equipment has sensors, like for example CO2 incubators, we provide sensors calibration certificates, and we also provide compatibility certificates with sterilization procedures, like very popular STERIS VHP, vapor hydrogen peroxide procedure. Okay, so let's explore on that second challenge. Big challenge for cell and gene therapy institutions, quality management system. So just to visualize this system is built with four main elements: quality control, quality assurance, quality risk management, and quality audits. And there are unique challenges with all these elements. I have listed the most important things here to remember. Quality control needs to become everybody's best friend, really, when you work in GMP lab. It's everybody's responsibility, consumes huge amount of time, and requires extensive trainings. It will be rather overwhelming for you if you are transiting from academic lab into GMP lab. And my tip to you guys, just be patient. Every experienced, even experienced academic scientist can become QC champion if that comprehensive structured training is provided. Quality assurance is higher level really of QC and focuses on preventing rather than only detecting those quality issues. And the people appointed in that quality assurance team must be very passionate about QC. And I would also recommend to source and hire super experienced individuals into that team with deep understanding of quality issues and risk. So next in line, we have quality risk management. And this is also similar as to quality control in a sense that it's everybody's responsibility. I remember working in a Covans that we had regular annual trainings to remind us how to recognize these risks to quality. And of course, the last quality audits, they can be external and internal. They can be super stressful, but they are very crucial to support continuous improvement to quality. And preparation to these external audits can be very challenging. And that's why we have actually separate webinar for you designated to audits. My colleague Dean will take you through the preparation for the audit and explain what to do and what not to do. I hugely encourage you to register and also participate in that webinar dedicated to audits. Okay, so let's talk in this slide about last big challenge associated with equipment. So product selection is challenging as we don't have a one golden standard for GMP lab equipment. Quality can vary between different manufacturers, suppliers, and many newly formed cell therapy institutions make wrong choices, especially those small companies, as lack of information or understanding. My advice is to look for experienced, reputable supplier which can provide you with all necessary info and advice. The second part, supply chain, can be also challenging. Therefore, flexibility in that supply chain is critical. That's the most important bit. And also many challenges are associated with product performance. And very often this need to be faced after purchasing of equipment. You know that realization that some certificates can be difficult to obtain, or problems obtaining correct services in the form of installation qualification, operation qualifications, or even lack of compatibility with certain cleaning procedures or with your unique processes. My advice to you is you need to really consider everything, and that ability to look even beyond in a future is needed here. The key is to have your processes developed, and knowing how the equipment fits into these processes is actually quite essential. Okay, guys, so again, to engage you and break down that presentation, the question number 2. So again, it's quite tricky as more than one answer could be possible, looking at it first. But I would like you to select again most prominent, important reason, only one answer. The only one reason for that documentation to play a huge role in GMP. The answers are A, allow to control employees' performance, B, it is crucial in maintaining employee safety, or maybe C, ensures quality systems are implemented correctly, and D, ensures traceability and ultimately patient safety. So I can see the answers coming already, thank you. So yes, so the biggest reason for documentation to really exist in a GMP is just to ensure that traceability and making sure we can trace, for example, if batch of the product is not as good quality, we have to trace it and identify reason for it, because this ultimately can determine patient safety. So the main reason for that documentation is to ensure that traceability and ultimately patient safety. Okay, now when we discussed main challenges, let's talk about the step-by-step equipment acquisition in GMP. And what should you do step by step? I must say, there's no golden rule for this process, but hopefully this will give you a general idea and help you to draw similar plans of action, more specific to your unique needs. So starting from 1, extensive research is always, and with everything really, extensive research is always a key to success, I think. And it is something we cannot avoid. My recommendation, don't be shy to ask questions. Look for different type of advice. Make contact with regulatory bodies in your country. Make contact with equipment suppliers. Do your comprehensive homework. Second step, which involves developing your unique processes, it's really closely connected with implementing GMP guidance. You need to literally define GMP for applications unique to your processes. In other words, as we said in the early slide, GMP is not a set of golden rules. It needs to be adapted to your unique applications and processes. The next third step is to identify all needed equipment and extensively research your options for equipment. Identify critical attributes needed, key documents required. This will allow you to also create your checklist as per step 4. Listing what is crucial for you in terms of complying with GMP guidance. Then translating this into equipment attributes. And for example, if the impact of the air purity is crucial to you, you have to make sure the equipment you're choosing is designed to produce less particles and comes with compatibility certificate to, for example, ISO class 5 and so on. It's the next step, number 5. You need to start contacting then different suppliers, equipment manufacturers. And by doing all the previous steps correctly, which we discussed on the previous slide, you will be well prepared now for the conversation with supplier, and you will know exactly what type of questions to ask. In the next step, number 6, qualification of equipment, and this is critical step. Make sure you choose very experienced provider for your installation qualification, operation qualification, and really ongoing performance qualification activities as well. Look carefully for complete design qualification package with end-of-line testing, certificates of analysis, and really good drawings. All these documents and many more, depending on the type of equipment, should be included. In the next step, number 7, it is actually your responsibility to start developing all those standard operating procedures we were talking about, designated to your equipment use, cleaning, maintenance, and so on. And I would hugely recommend to acquire somebody or have somebody with extensive experience in SOP development and writing, as believe me, this task is very time consuming and can escalate and it can get easily out of control even. The last step, number 8, is all about correct preparation for the very stressful audits and making sure you choose experienced provider again for this preventative maintenance. And this can help you to pass those audits. And as I mentioned before, we have prepared for you that separate webinar designated those audits. In the last section of my presentation, I will talk about how choice of equipment should align with key GM principles. The most critical question is how the equipment can support GMP compliance, right? Let's concentrate on that schematic box on the right side of this slide first. As you see, the cell and gene therapy GMP institutions need to implement the procedures and all necessary controls to align with key GMP principle listed on the left side of the slide. And doing so, they can pass external audits, become compliant, and be ready to manufacture hopefully that super safe and high quality product. But how individual key GMP principle, all those 10 listed here, should align with different equipment attributes? Well, I will explain that based on the example. Let's say you are looking for CO2 incubator, okay? And what should you look for? What type of attributes should be critical to align with these GMP principles? So first GMP principle is associated with a detailed step-by-step procedure and work instructions. So how this relates to CO2 incubator? Well, your CO2 incubator in this case should also come with easy to follow instructions, preferably with some references available to demonstrate it is used effectively in the applications already by different institutions, different companies, with documents certifying its suitability for intended use in form of all those validation protocols to satisfy installation qualification, operation qualification template. It should also come with comprehensive, easy to follow user manual, training programs if possible, easy to follow installation checklist, and of course, design qualification documentation, which is crucial, including those functional specifications, end-of-line testings, and drawings. Second GMP principle is associated with carefully following these clear and logical procedures. And to make this possible, your selected CO2 incubator should be easy to operate. The SOPs are much easier to create and follow if CO2 incubator comes with system not prone to errors or contamination. Daily setup use should require minimum time. And usability really plays a huge role as well. This includes simple to operate and read user interface, as well as operating ranges should be clearly identifiable. Next GMP principle require you to promptly and accurately document work for compliance and data traceability. So therefore, in this case, selected CO2 incubator should allow that data recording and display at any given time. You need to be able to obtain data, log in information, and those key parameters should be easy to verify at any given time. You should have ability to calibrate any sensors. The CO2 incubator should come with also material certifications for that traceability. Fourth principle requires you to validate the system to make sure it's performed as designated, perform as designed. In this case, CO2 incubator should be easy to qualify and calibrate. This involves IQ or Q compliance. To do so, reliable services should be provided from the supplier manufacturer. A system should be easily to calibrate, equipped with alarms and alerts, critical to provide for safety or power faulty situations. All sensors should come with calibration certificates as well as certificates for quality and safety. And a fifth principle requires you to design and build proper, which mean highest quality facilities and equipment. And again, for CO2 incubator, this means it should be designed to allow easy cleaning, installation, and movement. You should be able to move and store CO2 incubator, and this procedure should be provided by your supplier. High quality design means high resistance to fluctuations in temperature and humidity. It should be also easy to clean. Data for surface reactivity should be provided, as well as cleaning procedures, including that compatibility with popular VHP method. And most important, CO2 should have built-in features to combat and control and prevent contamination. According to sixth principle, you should properly maintain the facilities and equipment. So therefore, CO2 incubator should be again easy to use, clean, and maintain. This means CO2 incubator should come in terms of use with adequately defined utilities, air, water, lighting, and electrical supply, as well as electrical circuits identified and load specified. In terms of clean, as we already mentioned, it should come with recommended cleaning procedures and clearly defined materials. In terms of maintenance, it should come with defined service procedures and preventative maintenance contracts available. Also, it should come with list of all the spare parts available. As per seventh principle, you should clearly define job competence and training. Your CO2 incubator in this respect should be easy to train on, provided with comprehensive, easy to follow user manual. Training checklists should be available and easy to follow. The unit should be especially easy to train on for new employees. Also, customer support should be provided from the supplier with possibility for additional training sessions and specialists available when you have any troubleshooting. The system preferably should also come with user controls and authorizations if needed. And this one is my favorite principle, number 8. And because you must to protect product against contamination through good hygiene. And contamination, you know how important it is in GMP. The CO2 incubator specifically should be designed with great focus to combat that contamination. All the attributes are really crucial in this case. Validated and tested built-in contamination control features are paramount. Design of the unit should allow minimal particle productions, and surfaces should prevent of microbial contaminants to propagate. To allow easy decontamination and cleaning, the design of CO2 incubators should not have dead spaces, porous surfaces, or difficult crevices to access. And the materials in the unit should be resistant to cleaning agents. This should be tested by supplier, and a recommendation for this should be provided to you guys. And the CO2 incubator in GMP, also clean room specialist settings, should be able to withstand that cleaning by VHP, vaporized hydrogen peroxide procedure. And of course, gaskets and seals shall conform to standards for clean room. The ninth principle requires the design and build quality into the product. To do so, the manufacturer high quality product, to manufacture that super safe product, your CO2 incubator also should be built and designed to high quality specification. High quality therapeutic cells can be only grown, and I'm sure you're going to agree, in the high quality designed CO2 incubator. And many attributes need to be taken under consideration here as well. Some of them we already discussed with other principles, but additionally to that, customer service provided by suppliers should timely respond to your requests and concerns. Even product packaging should come with easy to understand labeling. And if required, the components should be sourced adequately to prevent shutdowns. And CO2 should come with calibration points easily accessible from outside the unit. And software revisions should also be available and controlled. And the last principle is, of course, associated with audits. They are very important. And it is your responsibility to conduct this planned periodic audits. And CO2 incubator should allow you for easy planning for periodic audits, and supplier should assist you in passing these audits successfully. The service provider by supplier should be able to provide PM checklists, should give you a flexibility in terms of different levels of service contracts, depending on your unique needs. And this service specialist should understand clean room requirements very well, of course. Good planning for maintenance visit is critical due to clean room processing timing. The supplier should be able to provide additional services like temperature mapping, plus help you with many other unique requests. Okay, so after going through all those principles, I have for the third and last questions. And it's actually about the equipment and how high quality built equipment supports production of high quality product in the case of cell and gene therapy, that high quality cell therapy products. And we have again 4 different answers. A, allows to detect quality issues much faster. B, it makes quality issues undetectable. C, allows to document less errors. And D, ensures quality issues are less impactful. Okay, I can see correct answers coming. Yes, you're absolutely right. If you have a high quality build equipment in the GMP settings, it will support your high quality production of that high quality products, because this will allow you to detect the quality issues much faster. And this is crucial. Detecting those quality issues is really crucial in GMP. Okay, so after giving you the example in CO2 incubator, I would just quickly like to introduce to you our Thermo Fisher Forma Stericooled CO2 incubator, which is part of our CTS product portfolio and globally used by many cell and gene therapy institutions, mainly due to the high level of contamination control. This incubator with external humidity system, a feature HEPA filtration in the main chamber to achieve ISO class 5, independently validated, incredibly effective 12 log sterilization cycle. Basically, all the features to minimize the risk of contamination, which is critical in GMP and for cell therapy production manufacturing process. So our proposition to gene and cell therapy institution accounts for many challenges we just discussed in this presentation. First, we provide carefully selected GMP-focused products. We have in our portfolio CO2 incubator. We have biosafety cabinet and centrifuge. To make that compliance with GMP for gene and cell and gene therapy institution much easier. Additionally, we provide documentation package needed to comply with regulations. And then we also providing compliance services. So as you see, we are one company stop really to provide every needed element for cell and gene therapy institutions to become GMP compliant. And in my last slide, I would like to just really briefly summarize our offering to complete offering to cell and gene therapy institutions. Our cell therapy system CTS equipment portfolio support that GMP requirements in every really step of equipment life. And this is starting from selection, installation, and operation qualification, then process qualification, and of course, ongoing use, services, requalification, audits through the equipment life cycle. Thank you so much, guys, for listening to my presentation, and I'm gonna give over to Jeffrey now and for questions.
Thank you, Chris, for a very informative section. So I think please feel free to type in your questions. And in the meantime, I have collected a few questions already. So maybe I can start with the first one. But anyway, please feel free to type in your question. So the first one is, so probably this gentleman must have seen or met many people before. So his question is why some people would need a CO2 incubator with basically minimum particle emission to the environment, but others do not. Will my auditor fail my facility for not able to monitor the particle emission of my equipment to the clean room environment? So this is the first question.
This is a great question, Jeffrey. And yes, CO2 incubators on many occasions, especially when it comes to that cell and gene therapy manufacturing production, if you produce that, for example, CAR T-cell therapy in your CO2 incubator, these incubators have to be placed in clean room settings. And those clean rooms are all about controlling the particles in the air, to filtering those different particles, because those particles actually can carry risk of contamination. So that's why we have to minimize the amount of those particles. And incubators these days can come with validation and special certifications and adaptations to minimize production of those particles in a clean rooms. CO2 incubators can be also be placed in those GMP quality control labs. And those GMP control labs are really different to those clean room labs, because in clean room labs you manufacture that therapy, so you have to have minimal risk of the contamination. But in a GMP QC lab, you actually develop and run different methods, processes to test safety of produced cells. So in this case, the rules are not as strict to control those particles. So the incubator very often doesn't require to be as strict. But when it comes to audit passing, yes, in a clean room, because you have to control and you have to provide the data for the particle emission and particle control and what is the particle amount in the air. And to pass those audits, you do need special adaptation to CO2 incubators, or special CO2 incubators which control themselves those particle emission. Yes, I think.
Yes. Yeah, thank you, Chris. This is a very, very detailed answer, right? So I have another question. I think definitely we have people, well, from different countries in the region. So this one, I guess, is from India, because the US FDA, they have the office set up in India. And the question is, what is the US FDA expectation when they are inspecting a GMP facility? So it could be a very broad question, but what would be your answer if in short?
Okay, so yes, it's very broad question, because obviously it's many requirements to pass those audits. But now how we're developing, how GMP labs develop, and what I remember working in GMP lab and creating those methods to be suitable to run in GMP labs. You know, a lot of equipment placed in those processes. A lot of this equipment have to have ability to control electronically, and this regulation is associated with a code of federal regulations, CRF, and this is called part or title 21. And this code is really part of the FDA actually, and is those regulations associated with electronic records and electronic signatures. And I know a lot of people ask these questions. It is important for equipment and systems to have special adaptations. And yeah, it is very important. It's required from manufacturer, from the GMP institutions, because you need to have those different processes and controls designed to ensure that authenticity, integrity, and confidentiality of electronic records and signatures. And we going into that era of recording all the data to having all the data in a type of electronic data. So all this have to be accordingly adjusted and controlled. And of course, FDA will really will be very important for FDA to monitor that. Okay, so
Okay, all right. So here is a question. So what is your suggestion on controlling the potential contamination from maybe the water reservoir or from the water bottle inside the CO2 incubator?
Okay, that's a good question as well. And working as a cell culture application scientist, of course, I have many questions coming to me, this type of questions for the clients and internally as well. So as I mentioned, as I talked to you about our sterically CO2 incubator, which is part of our CTS product portfolio, that incubator have external water source for that to monitor that and to have that relative humidity in your CO2 incubator. As you know, it is very important parameter of growing cells. So firstly, it is external because you don't have that water pan. So you don't have directly water pan in that incubator. So you automatically minimize the risk of contamination by not having that water pan in incubator. You have that external system. But even you have that external system and you have that water in a bottle, and is by different mechanism that water from the bottle is distributed into incubator, that external system have to be regularly also cleaned, disinfected, and we have actually sterilization cycle on that incubator, which helps you to keep that contamination risk under control. It helps you prevent that contamination. Of course, I remember working in GMP as well, every system having a water. And it's not just CO2 incubators. It can be, for example, I don't know, HPLC, high performance liquid chromatography. You know that system have to be flushed with water. I remember working with this kind of systems, the water had to be actually changed every 3 days on a regular basis. It had to be correct type of water as well. And this applies to CO2 incubators, that water have to be changed on regular basis. The bottle, even that outside humidity system have to be disinfected and kept really clean according really with all the procedures provided by supplier, in this case by Thermo Fisher Scientific, in our user manual.
Okay, so now there is another question. This one, I think this is a very common FAQ for people who are first coming to the GMP world, and that is, do we have any public available document or guidelines to to help us how to buy equipment suitable for a GMP facility? Is there any such a guideline publicly available?
Yes, there's so many different guidelines and different standards available, and they are usually available by looking simple and doing simple research by yourself, really, electronic research, internet research. It can be firstly really confused what kind of guidelines you should follow. I like really the ISO standards, which really they have, especially recent ISO standards, then really have the comprehensive selection of rules how equipment should be designed, especially when equipment goes into clean room settings, clean room facilities. And I personally preparing different materials even for Thermo Fisher Scientific, and writing materials about how this equipment should be designed. It helped me to understand it. So yeah, I would recommend ISO standards, and of course many other standards like FDA regulations and different parts of that regulation as well, reading them. And then other than doing your own research as well, don't forget that, of course, you have different regulation and I believe different regulatory bodies in your country as well, which you can, make contact with and have a discussion with those guys as well. And don't hesitate to contact, of course, manufacturer of the equipment, supplier like us, like Thermo Fisher Scientific, because we do have a lot of specialists in our teams. And we do are really happy to answer a lot of your questions and also those regulatory questions as well.
Okay, so I guess we could accommodate two more questions. So this here we have one questions about DQ, the design qualification.
Yes.
Could you elaborate more what is a design qualification and what should we look for in the DQ? And should that be somehow provided by manufacturer, or is something that should be prepared by the user?
Very, very good question. And it's really important for especially startup companies to actually understand what design qualification is. Design qualification should provide document certification that the design of the equipment will result in a system being suitable for intended use. And the design qualification documentation should really be provided by a manufacturer, supplier, in a form of different technical data sheets, different drawings, and this can be the drawings play very actually important role in it. The piping and instrumentation drawings, they have to be really as accurate as possible, and they have to be should be provided by supplier, and they are provided by us as Thermo Fisher Scientific. And there can be as well different functional specifications. There can be hardware and software, different specifications. And, even I discussed that with our global cell culture scientist yesterday, with our Mauri Kay. She's very experienced, and she mentioned to me even, the documentation even for some of our sterilization cycles, because like we have on CO2 incubators, we have so many validated independent tests performed, and this is also available to our customers. So everything what we really implement, all our features into our equipment and the great attributes into our equipment, we have really a lot of proof and validation and documentations as well. So that the design qualification is really super important. And definitely supplier like us should provide you with guidance and with certain different documents which are part of that design qualification.
Okay, Chris. So I think because of time, so this is the last question. We are 1 minute overrun. So the last question is, GMP requires computer system validation, CSV, of certain laboratory equipment. Does your equipment qualification protocol include the CSV protocol? So this is a very specific question.
Okay. So I remember when I work in a pharmaceutical company in GMP labs. I think I mentioned already another question. We had a lot of, in our process, we had a lot of pieces of equipment which played really significant role in those processes, and we call them critical equipment. And this, for example, when I was developing those characterization methods for those different biologics like monoclonal antibodies, especially those analytical methods, for example, high performance liquid chromatography was part of it. And we had system called Chromeleon, and I believe it is supplied as well by Thermo Fisher Scientific. And that system had to be validated in terms allowed us to electronically record every step of the procedure. So for example, if I logged into that system with my electronic signature and open that system and start particular instrument for my assay, for my method, all was recorded electronically step by step. So if I applied any changes to my protocol, any changes to that equipment in a process, everything was recorded. We called it electronic trail, really. And this had to be always available for regulatory bodies and for audits. And this was always a part of our data. And that's what I know. When it comes exactly to regulations and how this is set up as a regulations, maybe I don't have more deep, accurate knowledge. I don't know if hopefully I partly answered that question.
Okay, so maybe Chris, if you are able to get maybe talk to some other colleagues within the company. Say if you could give me back the answer, I can pass it
yes, of course.
To the people who asked the question. All right.
Absolutely, I try my best to find a more accurate answer.
Okay, no worries. But anyway, thank you, Chris, for waking up very early your time. I know you wake up at basically 5:30 a.m. Because you are from London, right? But anyway, thank you very much.
You're welcome, Jeffrey, and it was really, really pleasure.
Yes, and by the way, we will have another talk same time next week by another thermoscientific colleague who had close to 20 years working experience with Eli Lilly. So he will guide us through an audit journey in his talk. So please watch out for the invitation from our team. Thank you once again for your participation and thank you for your time. Have a very good afternoon and very good evening. Thank you all for your participation. Bye-bye.
Thank you, guys. Bye-bye.